Hi guys, this week we're going to talk about blood, chapter 10. And as a reminder, blood is the only fluid tissue in the human body. It's a connected tissue, meaning it's composed of two or more of the same cells with the same function. If you look at blood, blood is a dark red color, and that's when it's oxygenated. Anytime it's deoxygenated, it appears a dull red or purple. Typical pH of blood, 7.35 to 7.45, and this is especially important when we have proteins, enzymes in our blood that become denatured if this pH is out of range. Temperature of blood is higher than our body temperature at 100.4 degrees, and our body weight is 8% of that is actually composed of our blood. Here we're going to look at blood in a closer look. If we spin down blood in this container called a centrifuge, we can separate our blood into two major components. We have plasma, which is the liquid portion here in yellow, and we have the formed elements, which is this white layer and this red layer below. Plasma, most of that is composed of water. 90% of that is water. We also have our waste products like urea, uric acid, carbon dioxide. Our gases are here too, so our oxygen. Solutes, sodium, potassium, chloride ions, all found within that plasma. One component that we haven't really talked about much are plasma proteins, which help in the osmotic pressure of our blood. One that you want to know is albumin. And basically what this does is, if you imagine, I'm going to draw over here, let's see if this works. Okay, so if you imagine here's your blood vessel. Okay, let's get another color. And here are all of, oh, that's not the same color. Work with me here. Alright, so let's see if this works. Okay, it doesn't work. Alright, pretend this is a different color. Okay, these are our cells and our tissues down over here. Okay, so cells and tissues, and above we have our capillaries. Bear with me here. Alright. Naturally, we have lots of solutes surrounding and bathing our interstitial fluid, and also a lot of solutes here within our cells. Albumin is a protein molecule found within our blood, within our capillaries, and if we didn't have albumin naturally by diffusion, all that water in our plasma is going to want to go out to where there's a higher concentration. So think of osmosis, that specific movement of water from a lower concentration to a higher concentration. But because of all this albumin, this plasma protein, water is going to end up staying here within our blood to bring up that blood volume. All right, ugly picture, I know, but hopefully you guys get the point. Another thing I want to talk about is this formed element. 45% of our blood is composed of formed elements, and these are cells. The percent of red blood cells we have in our blood, it's called hematocrit, and that's this value over here. So hematocrit is percent of red blood cells. Another term we give to red blood cells is our erythrocytes, so you're going to see these interchangeably. Part of the formed elements is this white layer that you see here, and that's going to give rise to our buffy coat. Hematocrit, which is the red blood cell layer, is down over here. White blood cell layer is this thin white layer over here. What's in that are our white blood cells, which are part of our immune system. We also call white blood cells leukocytes, and we find platelets, which are used for blood formation or blood clotting. All right, so we just went over all of this. 55% is composed of plasma, and then 45% of our blood is composed of those formed elements. Here's a smear of blood. If you take a sample of blood, you'll notice here is what your blood will look like. You're going to have a lot more red blood cells than you do white blood cells. White blood cells are here stained in purple. And if you look at a red blood cell, okay, forgive the fact that I don't have enough space. Red blood cells have this biconcave shape. That doesn't really look like biconcave, so I'll make it more pronounced down here. If I can get this to work. Okay. Here is the first concavity over here, and here is the second concavity. Because we have this biconcave shape, it appears white in the middle, but all that is really is where these two layers of the cells are closer together. So it's not really like a donut, it just appears like one because of this concavity. Moving on, red blood cells are also we know as our erythrocytes. Their main function is to carry oxygen. And red blood cells itself can carry oxygen by smaller proteins found within red blood cells called hemoglobin. I'm going to move over here. So if you look at, here's our red blood cell, hemoglobin, our protein molecules just like that, found within our red blood cells. And each of these hemoglobin molecules bind to iron and they bind to oxygen as well. So this is how we carry all that oxygen is by that iron, oxygen binding to iron. If you look at one red blood cell, you have over 250 million hemoglobin molecules, which is why this is so important, is because all those hemoglobin molecules bind oxygen and give rise to the function of red blood cells to carry oxygen throughout the entire body. How we give rise to red blood cells is from a process starting from our stem cell line called hemocytoblasts. Red blood cells don't last very long. They wear out within 100 to 120 days. So about 4 months, max. One way we can stimulate our body to produce red blood cells is in the event that you don't have a lot of oxygen. So if we whole process of our body, we always want to maintain that homeostasis, have that normal body blood oxygen levels. Here, if we have little oxygen, we're going to stimulate our kidneys to secrete a hormone called erythropoietin, a hormone you do need to know. And erythropoietin is going to target our red bone marrow. Our hemocytoblasts, those red blood cell stem cells, are found here within our red bone marrow. So we're stimulating those hemocytoblasts to produce red blood cells. Once we produce more of those red blood cells, we're now going to increase oxygen in our body by the ability of these red blood cells to carry oxygen, and then we can once again regain homeostasis. If we don't have enough ability to carry all this oxygen, because we don't have a lot of those hemoglobin molecules in our red blood cells, this is called anemia. In the event that our hemoglobin molecules are deformed because the protein structure itself has some sort of deformity, this gives rise to what we call sickle cell anemia. Polycythemia is an abnormal or excessive normal of red blood cells. This can happen especially people living at high altitudes. If at high altitudes you have low amount of oxygen that you're bringing into your body, and because of that you're going to start producing more red blood cells in order to account for that loss of oxygen molecule. So overproduction of red blood cells gives rise to this polycythemia. Going on into this sickle cell anemia, what is that exactly? And as I mentioned, hemoglobin molecules found here within your red blood cells. Here is an amino acid sequence for hemoglobin, and what happens in patients who have sickle cell anemia is that their 6th amino acid changes from glutamic acid to valine. If we're not going to go into much detail about the polarity and the charges of these amino acids, but basically because we change from glutamic acid to valine, this changes the overall three-dimensional structure of the protein, and under normal conditions, what happens is these red blood cells, when they go through capillary walls, they can squeeze through the capillaries, and on the other side, near the venial end, they'll come out within the shape again. Now, people who have sickle cell anemia, once they enter into the capillary bed and they come out the venial end towards that inferior vena cava, what happens is that they don't restore this normal red blood cell shape. They have this sickle shape with this pointed edge, and because these red blood cells itself are not going to be able to carry as much oxygen, they're going to end up being disposed of within that spleen and within the liver. So there's a lot of protective mechanisms for this, especially in people in Africa who deal with malaria. Malaria will target your red blood cells, and it's a good thing for people who live there because once malaria targets your red blood cells, especially people with sickle cell anemia, these cells are going to end up going towards the spleen anyway and getting disposed of. So this decreases their ability to get infected with a malaria disease. So some advantages for people who live in those areas. Leukocytes is another formed element we have. This is found, it's also called white blood cells, and this is found within our buffy coat. Leukocytes are our body's defense against disease, and they can travel through our red blood cells, through our capillary walls through a process called diapedesis, meaning it's squeezing through those capillary walls in order to get to the site of infection. We have a couple of abnormal numbers of leukocytes that give rise to diseases. Leukemia, this is when the bone marrow produces an excessive number of white blood cells. Leukocytosis, this is when we have a white blood cell count of leukocytes above 11,000, and typically indicating an infection. We have 5 types of white blood cells that you should know: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And the way I like to remember these 5 types of white blood cells is the phrase, never let monkeys eat bananas. So N for neutrophils, L for lymphocytes, M for monocytes, E for eosinophils, and then B for basophils. Now, we're not going to go into too much depth in here about the different functions of all of these white blood cells, but you want to take a look at it in your book. But what you do need to know is that neutrophils are the very first type of white blood cells that enter into the site of infection. If you get sick or a cold, they're the first type of cells that respond. Lymphocytes are the types of white blood cells that are involved in our specific antigen response. So we have B lymphocytes, T lymphocytes. Monocytes turn into macrophages, and these become our antigen-presenting cells found within our immune system. Eosinophils are the cells that respond to parasites. So if you have a parasitic infection, you're going to have an increase of eosinophils. And then basophils are the ones that produce mast cells, which involve your allergic reaction. Another formed element we have are platelets, and these are derived from ruptured multinucleated cells. Alright, so if we imagine, and we have this large cell called a megakaryocyte, and this megakaryocyte, it's multinucleated, and eventually all of these smaller cells start pinching off from that megakaryocyte to give rise to all those platelets. So I'm not going to draw all of the layers, but just imagine this huge cell that's going to start pinching off to give rise to all those platelets. And we need platelets for blood clotting. Okay, so we'll get into that in a little bit. But what I want you to know right now are the different hormones that give rise to stimulating the growth of your red and white blood cells. Colony stimulating factors and interleukins give rise to your white blood cells, and thrombopoietin gives rise to your platelets. Alright, so I mentioned earlier hemocytoblast, stem cell that gives rise to our lymphoid stem cells and our myeloid stem cells. If we're going to start producing lymphocytes, that's from our lymphoid stem cell, but our myeloid stem cell gives rise to all 4 other types of white blood cells, our platelets, and our red blood cells. Okay, so, but what I do want you to know is that hemocytoblast is the primary stem cells that gives rise to all of our downstream red and white blood cells, and along with our platelets. Alright, next thing we're going to look at is hemostasis, so when those platelets actually come in. And hemostasis is the act of stopping bleeding for any time you have a break within your capillary or within your skin. And this occurs within 3 phases. We have the first phase being vascular spasm. Here, if you remember, within our capillary walls, we have smooth muscle in our tunica media. And because we have smooth muscles in our tunica media, we're going to start contracting them to help close off this gap right over here. That's the very first thing. And we want to decrease blood loss. Second thing we have here is platelet plug formation. Here, as we have this exposed collagen fibers, it has sticky ends. All the platelets are going to start adhering to those sticky ends to give rise to this platelet plug. We want to anchor those platelets in place, and so in order to do that, we got to have some sort of glue network. So I'm going to scroll down really fast, and that glue network is fibrin. This is that white layer right over here. And so we have to actually go through a cascade of reactions in order to generate that fibrin to make that platelet stick in place. And to do that, first we have platelets and damaged collagen, and that stimulates the formation of this hormone called thromboplastin. Circulating in our blood, we have a phospholipid called PF3. The one ion you do need to know that helps the formation of blood clot is calcium. And we also have other blood clotting factors. So collectively, all of these things will start initiating thromboplastin. Within our blood, we have this inactive plasma protein called prothrombin that's secreted by our liver. But in the presence of thromboplastin, which once again will only become activated because here we have damaged collagen and we have activated platelets that have been bound towards a site. Collectively, this is going to activate prothrombin to become thrombin. Thrombin, in turn, will activate another plasma protein called fibrinogen to form fibrin. Now we have fibrin, which can finally hold and clot and anchor all those platelets into place. Finally, here is that last phase. We have coagulation. Coagulation is that formation of all that fibrin that we just talked about, and anchoring all of these red blood clots anchoring all these platelets into place to form that blood clot. This entire process occurs within 3 to 6 minutes, and if that clot breaks away and flows within our blood vessels, that can be deadly, especially if it gets stuck into our brain. And that's called an embolus, so having that blood clot form. Now, by chance, blood clots can also form even if we have no break into our capillaries or in our vessels, and that's called a thrombus. A couple of deficiencies. If we have thrombocytopenia, this is a platelet deficiency leading to a blood disorder where someone cannot stop the bleeding, so it leads to uncontrolled bleeding. Hemophilia is another type of bleeding disorder where those normal clotting factors are missing, and leaving the person the inability to stop blood flow and blood from actually clotting. Large losses of blood have serious consequences. If you have over 30% of blood loss, that leads to shock and it can be fatal. 15 to 30% of blood loss makes you weak. We have two reserves in our body for in the event that you have a substantial amount of blood loss. We have our spleen and our liver that can expel blood to help restore blood volumes. Now the next thing we're going to look at are human blood groups. And human blood groups specifically have to deal with the presence or absence of surface antigens. Antigens, as we talked about before, antigens are surface markers that your body can recognize as self or non-self. Now, this is important when we deal with blood typing because in the event that you need blood, you have excessive blood loss, you need to receive blood, or in the possibility you can also donate blood to somebody who has a lot of blood loss. And so for that we need to type blood, meaning that we need to determine what type of blood that person has in order to ensure that somebody can receive or donate that specific type of blood. Agglutination is this process of blood clumping, and that's in response to antigens interacting with antibodies. I'm going to draw that for you really quick, which I once again stuck with drawing. But here, let's imagine we have a red blood cell. This is a type A red blood cell. And we have spherical surface antigens. And then to recognize this, so let's say this is yours, okay, your blood, and you have these surface antigens. Well, my body only recognizes an antigen that is triangular shape. And so I can recognize something as foreign by what we have, antibodies. And I suck at writing this out. Okay, antibodies. Just remember this is the antibodies. So antibodies are going to detect all of these antigens and cause them to clump and give rise to agglutination. We have over 30 common surface antigens, but because our class is only going to focus on our A, B, O, and Rh blood group antigens, we're not going to cover the rest. What you do need to know are universal recipients and universal donors. Basically, somebody who does not have any surface antigens at all, they're going to become a universal donor. If somebody has all the different possible amounts of surface antigens, they are going to be the universal recipient. The two that we're going to focus on are A, B, and O blood types. Let's say here this is a type A blood type, meaning that they have the spherical structure on the surface that symbolizes an A antigen. Somebody who has type B blood are going to have these pointed structures on the surface of their blood. And somebody who has type AB has both of these structures. If you are type O, you lack both of these structures on your surface, meaning you have neither of them. Somebody who has type A blood will not want anybody they would not want any donating blood donated from a person who has type B. And if you can notice, we only want spherical antigens. We don't want anything triangular. And because of that, somebody with type A is going to produce antibodies against type B surface antigen. So anytime this comes into our body, we're going to cause clumping and then get rid of it. Alternatively, somebody who has type B only wants triangular surface antigens, meaning this type of blood. So anybody who has spherical surface antigens, we can attack that, we can bind to that by producing antibodies that can recognize that spherical shape. So if this is brought into this body, we're going to bind to it through a process of agglutination and then eventually get rid of it by the body. Somebody who has type AB will have both surface antigens. So now think about, why is it then that they do not have anti-A or anti-B antibodies? Let's just stop and think about that right now. So they don't have it because if you had anti-A antibodies attacking your blood surface, then what's going to happen? You're going to cause agglutination of your own blood. If you have anti-B antibodies attacking your own surface, you're also going to have agglutination of your own blood. So we don't want any of that, meaning we don't have or produce any of those surface A or B antibodies. Now, if I don't have any antigens at all, why is it then that I would produce anti-A and anti-B antibodies? Yes, you are correct. So we don't have any surface antigens, meaning that if we bring any blood into this body, we're going to start attacking it because we only want blood that does not have anything. Very good. Okay, we're going to move over here really quick, and we're going to look at RH. What is RH? So when we look at blood types, we only focus on A, B, O, meaning you don't have any, and RH. All RH is another type of surface antigen. Here's my red blood cell. And so far we looked at the spherical shape, meaning it's an A antigen, a triangular shape, meaning it's a B antigen. And then we have here, let's say we have a square shape surface antigen, and this is going to be my RH antigen. Meaning if I have the surface antigen, I am RH positive. And if I don't have that antigen, then I am RH negative. Right? The difference between RH positive and A, B, and O is that you can have an O positive, O negative, AB positive, AB negative, A positive, A negative, and B positive, B negative. And what would that look like? So you want to draw it on your own. Let's see if you can get it. So let's draw first this red blood cell. And what would you say if I want to say I have an A positive blood type? And that's another thing. So we don't necessarily have to write RH positive. If we write A positive, we already mean that this is an A RH positive blood type. Or you can even write out the word positive. It's not necessary because this is already known by that. So if you have an RH positive blood type, what that would look like is you would have that spherical shape, and you would have a rectangular shape on the surface of your cells. As remember, this one was at RH antigen, and this one is at A antigen. Okay? Antigen. Now, let's do one more practice. let's say all we have was rectangles. What blood type would this be? Good. It would be O positive. We don't have any of those spherical shapes or triangular shapes associated with an A and B blood type. So we're O, but we do have that rectangular shape, meaning that we're positive for the Rh factor. So that is positive. Rh is another surface antigen, but it was originally found in the rhesus monkey, and so we have this association as we're related to them. So this can give problems, especially mixing those different blood types. And this is especially important if that mother is Rh positive or negative and having a fetus that is Rh positive or negative. So specifically here, let's draw a mother who is Rh positive, Rh negative, meaning that their blood type, they don't have any of those rectangular surface antigens. Now, if their first baby, so baby number 1, see if I can write this here. Cancel. I'll just write it out. So baby number 1 ends up developing that surface antigen for Rh, so they're Rh positive. Okay, but let's say the mother is Rh negative. Does the mother have antibodies for Rh? Right now the mother does. So the mother has a little amount of antibodies for the Rh antigen. However, if that fetus's blood enters into the mother is going to recognize this rectangular shape or this square shape as foreign, and because of that, it's going to increase the production of antibodies that are going to target this type of antigen. Okay, so over time, we're going to increase the production of antibodies that are going to target those square shapes. Now, this is going to be problematic if now the mother has lots and lots of anti-Rh, and has a second baby that is also Rh positive. Imagine if that mother's, and we remember, there's, actually, sorry, I didn't get into this yet, but antibodies, of our antibodies that we have, IgG antibody can cross from that mother to the fetus. Okay, so this is problematic if we have an antibody that can cross into this fetus and recognize this as being foreign. So what's going to happen is that the mother's antibodies can start attacking the baby's red blood cells. So to prevent this, mothers who have a first baby that are Rh positive, they will receive a shot called RhoGAM, and this will prevent the buildup of all these anti-Rh antibodies, therefore protecting a second baby who may be Rh positive. Alright, so kind of just sit on that, digest it, read over it a little bit more if that was a little confusing. So this is a little bit more into depth. Blood typing deals with cross-matching, meaning we're going to test for agglutination, whether it's going to be positive or not, and whether or not somebody can receive someone else's blood or not. The way blood typing works is that here we're going to take a vial. So let's say we have an aliquot of red blood. This is blood, an aliquot from a patient. Okay. And we're going to take a small sample of that. Okay, this is patient. We're going to take a cell sample of that, put it in a tray. Alright, so we're going to do 3 trays. Alright. And all of this in black, these black dots, all of this is a sample of blood. So we'll just say it's like 25 milliliters of blood. Alright. Now what we're going to do to this is we're going to test and make sure that we actually know this blood type. So to do that, we're going to add serum, meaning this is solution only of antibodies. So here we have an A antibody, meaning that it's only going to bind to an A antigen. Here we have an anti-B serum, meaning this is antibodies. It's a whole liquid composed of antibodies that are going to target B antigens. And then third one, we're going to have an Rh serum, meaning that's only serum that's composed of antibodies that are going to target Rh antigens. Now, this serum we're going to mix both of these together, and we're going to see what happens. If we see agglutination, meaning we see clumping, that means that we have that type of blood. Okay, so here, let's take this for example. We have a blood sample, so let's say we got it from this aliquot, and we mix it with serum. We got it from an aliquot down here of a solution only of A. We mix it together, we have agglutination, and that's because we have those antibodies interacting directly with antigens that have an A surface marker. Same thing here with B. We're going to add B serum to that blood. Here, this is actually, we start off, if you imagine covering this up, we don't know what that blood type is, but we see clotting, and that means that because we have agglutination, the blood type is going to be AB. Alright, so think about that for a little bit. Now, here we have the very first one, we have a sample of blood. This is now a new type of sample of blood. We have blood, we're going to add serum, anti-A serum. We don't see any blood clots. Okay, so we're negative for A. Here we have blood, we're going to add B serum, and we see clotting. What is our blood type so far? Good, so besides looking at here, this is a B blood because that anti-B antibodies are going to target and directly bind to any B surface antigens. Imagine here we have a third column. This is for our RH, and we have clotting. What would the blood type be then? Good, it would be B positive. So think about that. Go through this again. Make sure you understand that. Purpose, okay, so this we need to do blood typing before we do any blood transfusion. And so because this is very crucial that we know the blood type, we're going to first determine and ensure that we know the blood type of the recipient blood. We know the blood type of the donor blood, and then you want to select a donor's blood with both that A, B, and O blood type as the recipient and do a cross match. So you're going to perform this again. So you really want to make sure and ensure that the blood type is the correct blood type. Here is the list. You do not need to memorize it, but you should be able to understand it because this could appear on the exam. So here we have AB positive, meaning that you have all the A surface antigens, you have B surface antigens, and you have that RH positive. Because you have all these possible surface antigens, that means you can accept blood from any type of blood type. So you are the universal recipient. Lucky you. If you are O negative, okay, if you are O negative, you are considered the universal donor, meaning that you don't have any surface markers on top of you because, let's look at here, you don't have A surface antigens, you don't have B surface antigens, nor do you have RH surface antigens. So you don't have spheres, triangles, or squares, meaning that no one's going to reject you because there's nothing for them to attack. And because of that, you are the universal donor. So it's a good idea if you do determine your blood type and you find out that you are a universal donor, definitely give blood because look at how everybody can receive your blood. I'm an O positive. Kind of sucks because I can only give to half of the people, right? So only O positives, B positives, A positives, and AB positives. But hey, it's okay. Alright, looking at developmental sites of blood within a fetus, the spleen and the liver are the early sites of blood formation, and then eventually we have the bone marrow. And if you remember, if you look at long bones, okay, in children, bone marrow, red bone marrow is found within the diaphysis. Okay, so we have all that red bone marrow here. Red blood cell formation is found within the diaphysis. In the adults, we have red bone marrow here in the epiphysis, and this is where we're going to have our hematocytoblasts found, giving rise to our red blood cells. If you have any questions, feel free to email me, and that is all for lecture.